Okay, I think we're all online. Let's just wait a couple of more seconds for people to join. Right, so hi everyone. Welcome to BioC 2021 and this first long workshop from Christophe, Vanderaa and Laurent Gatto. And here we will learn how to run a single-cell proteomics data analysis using the QFeatures and SCP packages. And just before we start, a few housekeeping items. So if you have questions, you will see there is a Q&A tab in AirMeet. So please use that to ask any questions, and you can also upvote questions from other people. If you would like to ask a question live, you can also raise your hand, and we have the possibility to bring you on stage as well. You will also see there is a number of emojis available in the little smiley face where you can give feedback to the speakers as well. And finally, there will be a recording of this session available a couple of hours after the session is completed in the same place in AirMeet, if you would like to rewatch it or if you have to leave for some reason. So with that, I will leave the word to Christophe and take it away.
Yeah, thank you for this very kind introduction. And thank you all for attending this workshop. I'll start by sharing my screen. So here it is. You should be able to hear now. So yes, as Charlotte said, I'm Chris, and I'm accompanied by Laurent. On the left, you can see if you want to reach us, we're on Twitter, we're on GitHub. So any question you can ask them there later on. So also, I see already some of you noticed, but on the right. In the tabs, there's chat, question and answers, but there are also a few polls that we are that we have set up so that we can kind of monitor what's your background and knowing a bit like where you are at the moment. Meanwhile, maybe like just a brief summary in case you didn't follow Kevin's presentation about how to access the workshops. First of all, you have to go to the Orchestra app. Or maybe if Laurent or Charlotte can just put in the chat the link so that people can click on it. Sorry, I forgot to prepare it. But basically what you have to do is click on this link on to go to Orchestra. You will get directed to this little page. You will have to log in. You can either sign in with Google or with your email. I use Google, it works very well. I mean, I guess email, it works very well too. Once you logged in, then you will get to the workshop page. What I suggest you to do is to go in the search tab, put SCP, and then you will find the workshop. If you click on the link on the left, you'll get access to the website with the different there will be 2 vignettes available in the articles tab, and you can click also on the launch button. Then you will get directed to a new page where it will prepare the RStudio environment that you can use if you want to follow along. And yeah, have you hands on the package that I will show you today. Launching the RStudio can take a little bit of time, so if you can do it right now, I guess that's the best. So that once you have to get into action, that's already everything is already set up. Of course, if there's any technical issue, don't hesitate to put a question in the question tab. Okay, and with that, let's go. Let's first go with the first vignette. So I'll keep the screen on the right. So if some of you are a bit lost, they can still find it. But on the left, I will introduce you first. Well, I'll first give you an introduction about QFeatures and let's say more in general what is analyzing proteomics data before digging into single-cell mass spectrometry-based single-cell proteomics data. So basically, mass spectrometry-based proteomics data starts with a mass spectrometry experiment. Out of it, we run some samples, and out of it, we get some raw spectra. This is some kind of measurement that the instrument does. And those spectra are not informative as is. And so they have to be processed through some software that will perform, based on those set of spectra, they will identify from which peptide and in the end protein it comes from, and it will also perform quantification. And so the idea is we start with this. We have a list of features that are quantified across different samples. And when we say quantification, we immediately think about SummarizedExperiment, of course, because it's a bioconductor, right? Let me just see. Okay, very nice. I see that actually all people that have answered the poll are familiar with SummarizedExperiment. So I'll go quickly through that. So most of you probably know that SummarizedExperiment, while the core information is contained in the data matrix that you can retrieve using assay, the assay function. But then to each row that corresponds to different feature is associated some metadata, and to each column that are associated to samples there's also some metadata available. And so so now that's just to give you an introduction. What does QFeatures? Well, QFeatures, it will help you construct from this identified and quantified spectra. So this very, let's say, crude data, it will help you go through the process of building up the protein data, because actually when running an MS experiment, you digest proteins into peptides and then you collect PSMs. Well, what you want to do here computationally is to go the other way around. From the PSMs, try to re-estimate the peptides and then go to the proteins. And this process of going from many features to lower number of features, reconstructed protein data, is called aggregation. And this is represented here by this very simple schematic. Suppose on the left you would have your summarized experiment. So you can see you have many features, and some features might aggregate to a single peptide. And so this middle box represents the peptide data. Then for biological interpretation, we are more interested into proteins. So from the peptide data, we perform again an aggregation and get the protein data. So several peptides can aggregate to a single protein. So QFeatures is actually built upon multi-assay experiments. So maybe some of you know it. It allows to have several summarized experiments inside a same container. The advantage of QFeatures is that not only it will contain those different assays at different levels, but it will also keep track of the links between the features. For instance, we will keep track that this protein came from those 3 peptides that came from those 6 PSMs. PSM, sorry, stands for peptide to spectrum match. And so we start off by running, loading the QFeatures object. What we do then is, in this case, I just show you an example data, what we call feat1. And you can see feat1 is a QFeatures object that contains one assay. So one summarized experiment that contains 10 rows, so 10, in this case, PSMs, and 2 columns, so 2 samples. This assay in the container is called PSMs. So now we can dig a bit into the data. What does it contain? So a QFeatures has some cold data, also some cold data associated. So for every sample, it will have, eventually, if you provide it, it will have some data. So in this case, we have one variable associated to samples, which is the group. We have group, sample 1 is from group 1, sample 2 is from group 2. Okay, when we have a QFeatures object, we can also extract the assays, so the summarized experiment, because we want to do some processing on it, or we want to visualize it. And you can do this by using the double bracket. Either you can say, okay, let's take the first assay, or let's take the assay called PSMs. This is doing exactly the same thing in this case. And you can see we retrieve a summarized experiment object. Of interest is to get the quantitative data, and you probably know that we can do this when we apply the assay function on the summarized experiment, so after extracting it from the QFeatures object. But we can also retrieve the feature annotations using the row data function. And then you can see we have different annotations available. For instance, so for each PSM, we have different peptide sequences. You can see there are here, some are redundant. We have proteins, you can have predicted protein location, and you can have some PSM specific features. So for instance, p-value, the p-value, the confidence we have in the identification of the PSM, or whatever other variable is here. It's a mock variable going from 1 to 10. Okay, And so now feature aggregation, this is probably the main, the core principle of QFeatures, is doing this aggregation, building back the protein data. So let's do this first, aggregating PSMs to peptides. And so what we do is we use the aggregate features function that takes the QFeatures object. It will ask for, okay, which assay in the QFeatures do you want to aggregate? In this case, we only have one, but still we have to provide its PSMs. How will we call this new assay? Because it will create a new assay with aggregated data. It's called peptides. We will do this based on the peptides sequence, and we use the function call_means. And so basically, if we have several features we want to aggregate, how do we aggregate this? Well, we do it using call_means. Maybe a little exercise at home. I mean, I won't go too much into detail right now, but you can redo what aggregate features does by extracting separately for each peptide the corresponding data and see that indeed we are just applying. I mean, QFeatures automatically applies call_means to each peptide. It will also combine, well, when aggregating, there's some raw data I show you, but it will also transfer the raw data to the aggregate feature. It will only do that if the variable that you are looking, well, if the variable is common to the peptide. So for instance, sequence, well, I mean, it belongs to peptide, so it was common for all the different PSMs belonging to the same peptide. Protein as well, and location as well. However, remember there was this p-value that we showed, and the var, the mock variable. Well, those were specific to PSM, so those are lost. They are not transferred to the peptide data. And here we have a new variable that was added. It's called .n, and this represents the number of PSMs that were aggregated to that peptide. So we had 3 PSMs for the first, 4 PSMs for the second, and 3 PSMs again for the third peptide. Okay, so we did this PSMs to peptides, but as I said, we are interested into proteins. So what we do is we do a second aggregation to proteins to get the protein data. And maybe that at this moment it might be interesting to show you. So maybe I give you here the little overview. So now we have 3 assays. We have PSMs with 10 features. We have peptides, only 3, and then proteins. Now that's only 2. And we can also have a little look at how. So I've run already the vignette. I can show you here. So again, it's exactly the same, just what I showed from the compound vignette. But we can also plot the feature object, the QFeatures object. And so if we do this, yeah, it doesn't like that my screen is. Sorry if it's all a bit compact. I'll remove this screen on the right once I'm done with this vignette. But yeah, and then of course if I don't put. Okay, and here we see this is a graphical overview of our QFeatures object. So we have the PSMs that are aggregated to peptides given by the directions given by the arrow, and peptides go to proteins. Now something also very interesting is subsetting of QFeatures. And maybe to show you this subsetting, the easiest thing is I made a quick drawing just before this. So there is QFeatures, you can subset it in 3 ways. So therefore there's a 3 index subsetting possible. And so if you look at here, so you can either have the first, second index, and the third index. The first index is will subset based on features. So you can see if, for instance, you're looking for that specific PSM, it will extract all the information from that assay for that specific feature, but it will also recognize that it's linked to other PSMs. So the peptide is linked to PSMs, and it will just collect all of it. Then you have samples. So samples across, because we aggregate, the samples are the same. So if you extract from one given sample, it will automatically recognize that those samples are the same, and it will extract all the information available across the different assays. And then if you want, you can simply access just an assay and say, let's say, if we want third assay, we will extract that assay. So how does that look? Into action. So we have here, we have our QFeatures object, the same as above. We take protein A, well, whatever protein that is, and you can see that in the proteins, we now have only a single protein. But that protein was linked to two peptides, and those 2 peptides were linked to 6 PSMs. And so we collect all of this, and we can easily retrieve this QFeatures data. So that's let's say, simple filtering. Then we're also interested to filter based on some metrics. And metrics usually are stored inside or some data stored inside the raw data. We want to filter the features. And of course, this function is then called filter features. So we can provide it the QFeatures again. Basically, any function from QFeatures will expect a QFeature function. So that's always the same. And then we can provide it a function. Remember, for the PSMs, there was this p-val function, this p-val variable. Well, maybe we want to keep only PSMs that have a significant p-value. And this is what this command does. So we keep only significant p-values. And then here you can see we are left only with 4 PSMs. Peptides and proteins, they are removed because they don't have this data available in the raw data. This is one example. Another example is filtering on mitochondrion and saying, okay, we want to keep only proteins that are predicted to be located in the mitochondrion. And in this case, maybe we don't want to keep those. And every time we have different filtering. This is the introduction about QFeatures. So here I just scrapped a little bit the surface of what all QFeatures can do if you are interested. So other functions are, and we will see it later, other functions are, for instance, we can do all sorts of processing like normalization, log transformation, imputation, filtering based on missing data. And yeah, if you want to have more information about this, I suggest you to have a look at our different vignettes that we built and that we provide along with the QFeatures package. Okay, maybe I'll show a last time this screen, because I hope now you are quite set up with the RStudio. I'll give you maybe like 5 seconds to memorize it because I won't show it again anymore afterwards. And so now what we are going to do is I'm moving to the second vignette. Oops. And the second vignette is more about single cell. So now we will still use QFeatures because the second vignette is heavily based on QFeatures, but we now focus more on single cell application. And yeah, so single cell proteomics using mass spectrometry is actually something very new and quite recent, thanks to recent technological advances. I won't go into details. There are different advances that came out lately. In this vignette, we will focus on one technology. Although the workflow can be adapted for all the other existing protocols, but here we will focus on one, and that protocol is called Scope 2, and Scope 2 was published by Spect and Al in this year. And here I will give you a brief overview that might give you also a bit an idea of how mass spectrometry experiments go. I mean, by this I mean this is very similar to like bulk, but here the advances are that the technologies were adapted for getting very fine measurements, because single cell are just very low amount of material. So the experiment starts with isolated cells. I mean, you isolate as you wish, but the idea is you start with single cells. From those single cells, you extract the proteins because you want to measure them, and those proteins are then digested into peptides. For each cell, you label the belonging peptides using a given label. In here, this case, we call it a TMT because that's the technology. So you apply a specific label, and once you did this for a certain amount of cells, so here it's up to 16 cells, well, once they're all separately labeled, they pull together. Once they are pulled, we put them through a chromatographic column because they will separate the different peptides based on their affinity for the column and for their mass. At the end of the column, they are ionized through electrospray, and the ionization makes them analyzable by mass spectrometry. The mass spectrometry, what it will create, it's a tandem mass spectrometry, so it will create two runs of spectra. So in the first run, it allows to further discriminate between the different peptides. So a given peak corresponds to one peptide. It will be selected and then sent for second round. And the second round here, the pattern that you will get will give an idea of what is the sequence. So those spectra are stored in raw MS files, and as I said, those are not informative as is, but they can be sent to a software that will do two things. They will take one part of the spectra and try to match it against a database, and this will allow them to retrieve. Well, it will allow the software to retrieve the peptide sequence. And on the other side, there is another region where the intensities that measured are depending on, are given by the label. And so thanks to that, we can do some quantification and deconvolute, I would say, or demultiplex the pool that we had in the beginning. And so have a quantification for each cell that was labeled. In the end, what does it lead to? We have each spectra corresponds to a spectrum, and when it's matched, we say it's peptide to spectrum match. So we have one row for each feature, and then we have one column for every label that was used, so for every cell. And this is just the beginning of the journey, as I just explained with QFeatures. What we then want to do is to reconstruct the protein data that will be used then for downstream analysis. And this process is not trivial. There are many steps. So there are quality controls, there are normalizations, of course, aggregation, as I explained. And so this is what we will see later on. But to be able to do this, and by using QFeatures, we had to not really adapt, but think about a conceptual data framework. And so we came up with the SCP data framework. And it looks like this. So we have our data framework, which is a QFeature. And within the QFeatures, we don't have summarized experiments anymore. We're now using single cell experiments. So it's a subclass of summarized experiment that provides a nice interface with existing single cell methods provided by the Bioconductor community. So no summarized experiments, single cell experiments. The other difference is that now the PSMs, because I said you can analyze up to 16 cells, but that's basically nothing in a single cell experiment. So you have to do multiple batches of acquisitions. So now each batch of PSM data is acquired in a separate assay. So we can have as many as PSM assays as they are batches. And for the rest, the structure remains the same. So we have, once we thought about this conceptual framework, we thought, okay, let's now provide some utility functions through the SCP package that will facilitate the analysis specifically for single cell application. And so the key function here is read SCP that will allow you from standard data tables create a standardized QFeatures object. But then you have also other functions that we will see later in this vignette. So now, okay, maybe a little word on how to load the data. So I was talking about standardized tables. Well, we actually have two tables. We have either the feature data or the sample data. So here in the vignette, you can find a lot of, I have formalized this all, but I think one image is better than just text. And I just made you this image here. So here on the left is an illustration of a data table as it would come out of a pre-processing software. So what you have typically is you have feature metadata, all sorts of metadata that are generated when doing the identification. For instance, peptide sequence, the charge of the ion, the mass of the peptide, and the confidence we have in the matching, well, all sorts of those kind of information. Then we have the quantification columns. So remember, we have per run, we can have up to 16 samples. So we can have up to 16 columns, quantification columns. And then finally, we have the MS batch. So basically the name of the file where that PSM was retrieved. And you can have several files. On the other hand, so that's for the feature data, for the quantitative data and metadata. Then you also have the sample annotations, because I mean, it's nice to have samples, but sometimes we want, well, I mean, in downstream analysis, we want to do more than simply having quantitative data. We want to maybe do some differential expressions, so having conditions, or we want to keep track of some batches during processing that might influence the quantitation. So we have sample annotation, and in this case, what we have is we have two required columns. First one is the MS batch. That's exactly the same as here. So each sample belonging to a batch. The other one is having, okay, in what column do we find that sample? So for instance, we have this corresponds to one sample. Well, to that sample, we have given some data. And what read-ECP does, it's take those two tables and it will combine them in a QFeatures object. And here you can see again, we have all different assays formatted as single-cell experiments, and we have associated the call data that was taken from the sample annotation. And that's one of the main features of SCP. So now let me go back here. So yeah, that's basically what I just explained. Maybe we can quickly look. So for instance, here we have the different channel names, so the different labels. Well, they were quantified in a channel name, so it's called reporter intensity 16. For instance, here you have the quantitative data. Here you have some examples, as I said, of the raw data. And then you have the different MS batches. And then the sample data, here you have some examples. So for instance, of course, you can have the sample type. Here in this case, there are two types, or different types of samples, but they were the authors were analyzing monocytes against macrophages, but they had also other kind of information about batch effects. So for instance, chromatographic batch, the salt day, the type of digestion, and yeah, you name it. And so in the end, how it looks, read SCP, it takes the two tables. So this is the feature table. Here is the sample annotation. You need to ask him, okay, how can we combine? So how do I know what is a batch? It's based on the raw spectral file names. And then how do I know where the quantification columns are? It's in the channel column of the annotation. And yeah, that's basically it. In the end, then you get, so what comes out of the read SCP is a QFeatures object with, and in this case, 4 assays. Okay, let's go. I mean, I quickly go over this. It's because we're not using it today. I just want you to know, okay, this exists in case you want to apply this kind of, if you want to apply our package and our functionality to your own data. But what we will do today is use data from the SCP data, which is another package where we provide already formatted data. So here, if you load SCP data, you can find different data sets, and you can, for each data set, you have a loading function. So for instance, if you're interested by the data set called Zoo 2019 EL, you just call the function and then you get your data ready to process. So we're here, for instance, with 62 assays. All right, it's now time for the more, let's say, hands-on, or at least more the practical part of the workshop. I suggest we take maybe a little, maybe not a break, but like a break for questions. I see there are some questions in the question tab, so maybe I'll take the time now to answer them. So I see, for instance, Danielle says, where do the protein reference sequence come from? We work with genetically diverse heterozygous mice that may have both a reference and an alternate protein sequence. Okay, yeah, so this matching, this identification is done, typically what people do is they download the UniProt database where they have all sorts of protein sequence. Since during the experimentation we use a specific, well, it's trypsin. We know where trypsin digests, and so we have different peptide sequences. And so based on this, we can have a set of predicted spectra, and then we can match the theoretical from the database and with the actual spectra that you measure. So I guess, I mean, I personally haven't tried this yet, but what I would do is, if you expect your proteins to have different, how would you say that? Yeah, divergence, is to include in this UniProt database, maybe like include several few fields with new proteins that are adapted from the original one. That's how I would do it, at least. When I tried to code read SCP, I got an error in as data frame.metatable argument metatable is missing with no default. Okay, that's an issue. No, that's okay. So Max, can I ask you, did you try to run this through the through the RStudio provided by Orchestra, or did you run it on your local machine? Because definitely this error is because you are using an older from RStudio. Okay, I mean your RStudio or the RStudio of Orchestra? Because the thing is, this is an error, meaning that the wrong package version is used. It's yeah, okay, your log. Okay, maybe I'll just put in the chat what you should set. So what I suggest now is you using the development version. So from oh wait, maybe, yeah, no, I'll type it in the chat. So I suggest you to use the development version, because yeah, lately there were quite some changes. So during the new Bioconductor release, there will be quite some new changes that will be made, and namely we refactored a bit the names to make them more clear. /scp. Okay, in the chat I just put a yeah, I've put a command line that if you run that locally, you should be able to run. But make sure that something that's important is you have to run on the development version, otherwise you will run into other issue. Otherwise, if that's the problem for you, what I would suggest is go through Orchestra. Yeah. Okay, I don't know. Are there any other questions? Okay, well, give me 1 sec. All right, then I suggest we move on to the exercise. So here I will switch to, oops. I will switch here to the vignette. So if you want to follow along with me, what you can do, I mean, if you're in RStudio from the Orchestra, so make sure to so it should look like some. I mean, it's maybe a bit small for you to see. You should have from your browser access to Orchestra and have your RStudio initiated. What you can do normally, if you launch the thing, you get this page, and then if you go into files, wait. You get here. If you go into in this files tab, you can go to vignette. You go to VO2 SCP, and then you will get this thing open. I suggest we move immediately to line. Where are we?
Christophe, I just want to say I just added a poll to just check whether people are up and running on the Orchestra or Studio instance.
That's
and also to make sure that people don't copy your URL, because then they will throw you out. So make sure to generate your own instance. Don't copy this address.
Okay, I just deleted it, so I make sure people cannot copy it. Okay, yeah, thanks a lot for the poll. That's good. So if you could go to the poll tab and answer, so we know who is not ready.
Looks like we have 8 yeses and no nos.
Okay, perfect. Well, then let's do it majority vote. Who says words to consent? Let's say. Maybe so here before we start. So I was saying, we are working with the Scope 2 technology, well, on data that was acquired with the Scope 2 technology. And what we are actually going to do is trying to reproduce part of the data analysis that the author made. So they provided some R script. They basically built everything from scratch. It was quite hard to read. I mean, the whole purpose of the paper was first to show that the technology worked. And so we took the effort to kind of standardize the script and show here that actually performing SCP processing is not that hard and doesn't require that much processing skills as long as you have the correct tools to do it. And so here, this is an overview. So you can see my screen. This is an overview of the pipeline that we are going to follow. There are a lot of steps, and so I want to, maybe it's a bit disappointing, but we won't have time to go through all the steps. So I will select just a few ones that I think are might be interesting to you. Other ones being more common, you can just follow the vignette afterwards. And so you can see I've colored different boxes depending on if it's blue, it means that the function is already present in QFeatures, and orange means we have implemented it ourselves. We have implemented it in SCP. And so you can see that actually for the moment, most functions simply rely on existing standard proteomics functionality, and there are only a few that required very specific or more specific single-cell dedicated functions. Okay, let's start. So first thing that we do is we import the data. So, what I needed to do also is, sorry for that. I need to load my libraries. So that's at the very beginning of the script. So we need SCP, of course, SCP data to retrieve the data. SVA is to do some batch correction afterwards. And then we have tidyverse. It's to mainly to get access to ggplot2 for plotting. So while this is doing, I'm going to scroll back. I hope you're not dizzy because of me. And we go back to line 354. And so it's still loading. So maybe I can explain what is happening here. So here we are loading the whole Scope 2 dataset. So it contains, you'll see it will contain 177 assays. That's so different MS runs. In a total, we have more than 1,000 cells. But here, what we are going to do is to use the LCB, only data coming from the LCB3 batch. That's just one batch with a lot of cells and that are representative of the whole data set. So before we start the exercise, maybe I'll, if you follow, so now I'll suggest two things. Either you want to follow and do things yourself, then I'll suggest you to go through the vignette. Or what you can do is through the vignette, through this vignette in RStudio. Or what you could do is simply follow my instructions from the compiled vignette. And here, so I've put some exercises. Wait, let me just, go back here. Here, I put this to the right, and you will see we have a few exercises. For instance, here, I put exercise, and you have the solution underneath. If you click on the square, on the triangle, sorry, then you get the solution. And for some exercises, there are even hints. So for instance, here you have different hints. Again, I won't go through all the exercises. If you want to make them all, I mean, the vignette should be compact, descriptive enough to be able to run them yourself. I will go just through a few representative examples. Another thing is that, okay, running the whole vignette can take some time. If you don't want to run the whole vignette again every time you make a mistake, maybe the easy thing to do is to to store the current data into a temporary variable, and then once you make a mistake, you can revert your changes and go back here. Meanwhile, it's still not loading. What is happening? Let me check the console. Oh, okay, sorry. I had to. So here, because SCP data is using Experiment Hub, there is some caching performed. So what you have to do is put yes when you load SCP data. And then now we're good to go, line 354. So now it's retrieving, you can see, so now it's automatically, it's going to Experiment Hub, it's retrieving the data and it's loading it. It can take some time because it's a big data set. Okay, it's taking quite some time, sorry for that. If you want to have a little drink, it's probably the right time. Excellent. Okay, so now it retrieved the data. You can see all the different assays. What we are now going to do is, so remember I talked about the three-way indexing. So now we're indexing on the third index for. So we are subsetting for assays that match to this batch. We run this, and then we can. Okay, maybe let me introduce the first step. So first step is to do the PSM quality control, so feature quality control. There are different metrics that we can use. For instance, here the first one is based on the number of features. So you can filter on failed runs because they don't have enough features. I'll show immediately the solution. You have a DIMS function that allows you to retrieve the number of features per assay. You can plot those assays, so let me do that. Oops. Up. Here you can have, so using ggplot2, so no visualization will be performed throughout this vignette using ggplot2 because I think it's nice. And you can see that here the distribution of the number of features per assay is quite high. So we have, I mean, for proteomics-wise, it's proteomics-wise, single-cell proteomics-wise is quite high, more than 3,000 features. So all assays seems to have run, so that's excellent. What we can do, we can also filter based on the false discovery rate. It's a bit technical. I'll just skip through it, but you can. So Max Quant, it's one of the software to do identification. It outputs some confidence, so posterior error probability for the match. And what the authors here wanted to do is they wanted to filter based on the FDR, so you can convert probabilities to Q values, and this is done using this function. Don't worry if the details are not very clear. I think the vignette is well explained, and otherwise you can also always have a look at the documentation, which works well. Here I propose a little exercise. So we compute Q values for PSMs, but we can also compute Q values for proteins if we group them by protein. So I'll just run this. And then, yeah, we can also show the distribution of those different metrics. So the idea, why are they doing this, is because filtering on probabilities is too stringent. It's better to filter on Q values. And so you can see, indeed, it's less stringent. There are way more significant features when we do this on Q values. And so what we do then, so actually it computed those Q values, and it automatically stores the computed Q values in the raw data of the corresponding assays. That's very handy because then, if you remember from QFeatures, we can use the filter feature function. And now our Q values are automatically accessible. And see, like in here in one line of code, boom, we can simply filter the features that are, how should I say? The features that have an FDR below a given threshold is 1%. So that's pretty handy. And if we look here, so we still have now we have only 24 assays from the LTB3 batch. Oh, and I should have showed this before, but if you have if you have looked at the thing before, wait, maybe okay, no, sorry for that. But you can see that some features were removed because they were not significantly matched to a peptide. We can do the same for contaminants. Here again, I'll just show you. So based on the protein name, some contain either matched to a reverse database or they are matched to a contaminant protein. And so those proteins, we don't want to keep them, so we can immediately remove them using, again, filter features. We can do the same with filtering out noisy spectra. This is based on a metric that's called the PIF, parental ion fraction. The higher it is, the purer the spectra, so the better it is. And so we can, again, using filter features, provide them the QFeatures object, and then providing the function, we can filter only for PSMs that have a purity above 80%. And so here now, okay, now I went a bit quickly through that because I think I'll say I think it's pretty boring. But here now, I think this is important because this is a new metric that the authors implemented specifically for the single cells. So something you have to know is that the QFeatures object, the scope 2 dataset has different types of samples. As I said, they want to compare macrophages against monocytes, both acquired as single cells, but they include also other internal controls. And for instance, we have blanks, so samples that were acquired as if they were single cells, but they are no cells. There are, for instance, references, a reference channel. So it contains a little bit more. It contains like five single cell. Well, it's not single cells. It contains 5 cells used for reference normalization. And then we have, and that's quite important, a sample called carrier. So in that sample, there are 200 cells, and this channel is used to boost the identification, because actually in one batch we then have much more material, and the more material you have, the easier it is to identify a spectrum. So they included this. But actually they thought also like, okay, this carrier, it's known to have many more cells. So if we compare the quantifications for carrier with the quantification for single cells, well, we know that we have some expected level. So if we have one single cell and we have in one channel, and then we have 200 cells in the others, well, we expect that this ratio is about 0.5%. And using this information, they wanted to compute the ratio between single cell and the carrier, in order to see, okay, if some features were badly quantified, let's say if they are, and we'll see some of them are way too high than expected, well, there is something that went wrong, and so those we need to remove. So to do this, I mean, either you can go into the data, take the different bits yourself, or you can use the compute SCR function. And so compute SCR, that's the exercise. That's the first exercise. So compute SCR, it will take the QFeatures object. It will ask you for which assays you want to compute an SCR. It will also ask you for a pattern. And so the pattern, how does it know what sample is what? It will look into the call data. So you need to tell him what variable in the call data gives you the sample annotation, the sample type. Well, I mean, now I give part of the solution, but there is a variable called sample type. And then you need to provide him a sample pattern, saying, okay, which columns are samples, and you need to give him a sample, a carrier pattern. So, and telling me, okay, which carrier is what. And then it will automatically do this ratio computation and store the result in the raw data of each corresponding assay. So now I'll let you work a little bit. I'll go through the hints successively, but I want you now to compute the sample to carrier ratio, which is a very informative QC metric for PSMs. just as a reminder. I mean, I'll let you work. Maybe I'll be speaking, but you're not obliged to listen to me. But here I'm just going to show you how to read. I mean, probably most of you already know how to get to documentation, but just question mark, name of the function. And then on the right, you can find all the documentation. There are some arguments that are required, other that are not. But I'll let you. So at this moment, maybe something that is interesting for you to do is, don't forget, is do TMP and then SCP. And so like this, what happened is that you stored, you kind of saved your data in a temporary variable. And now if you mess up with your data set, all you have to do is do SCP, reassign SCP to this temporary variable. No, the other way around, sorry. Yeah, that might avoid some annoyance. Okay, let me maybe go into the first hint. Yeah, so actually the first hint I already gave it. So you need to provide to say, okay, you need to tell compute SCR, compute SCR how to know which sample is what. And this is done by looking at the call data and which variable of the call data do you want to look at. And this you can actually, I give already part of the answer, it's through callvar. So which variable in the call data will give you the annotation. Okay, I'll maybe go a bit to this. Oh, no, it's already a solution, sorry. Okay, I write already here. So compute SCR expects the QFeatures object, which assays, so you can target select specific assays using the I argument. Here we say names of SCP, so we want to supply it all the names. The callvar, I told you, it's sample type. And now you need to provide a sample pattern and a carrier pattern. You can also provide the sample function in case you have several samples. In this case, it's the mean. And you can do the same for the carrier if you have several carriers in the same batch. But in this case, I will just ignore this and make things easier. So now the idea is, what do you put as pattern? The easiest thing to do is we look at here, sample type. And we see that the carrier, it's actually called carrier. So let's just use carrier as a pattern. When I say pattern, is of course, it's using the regular expression patterns. And so we have also, we want to use a sample, so macrophages. So I'll put macro, it will match macrophages. Or it will, we want monocytes. And so mono will fit mono, will match to monocytes. And I will also include blank. And so and that's actually the solution. Oh yeah, no, sorry, and then I forgot one. So as I said, you compute the SCR. Those computed SCR, they will then be stored in the row data. So we need to provide a new name for the row data. And in this case, so the row data name that we provide, we'll call it mean SCR, because we compute the mean over the different samples of the sample to carrier ratio. And if we do this, oh, wait, no, I didn't want to do that. Oh, well, okay. So I run this. And then that's it. We have our SCR computed in our row data, and we can now filter on it. But before filtering, maybe let's have a look at it. And so what we will do is we will get all the row data, and getting all the row data is performed using R-bind row data on the SCP object. Which row data do we want? It's from all these assays, and then we convert. So this will return a capital letter data frame. We convert it to a data dot frame because that's what ggplot wants, and then we just plot it. So yeah, and here we got it. This is our distribution of the mean SCR for every PSM in the dataset. And we can see, so the dashed line I show is the expected 0.5%. So on average, it's a little bit higher than expected. This is maybe because, or probably because the in mass spectrometry, the quantification is not fully linear. But you can see here we have like 10%. So where the signal of the sample is 10% lower than the carrier, that's way too high. We're expecting 0.5%, not 10%. And so the authors have arbitrarily put a cutoff here saying, okay, all that is above this means that there is something wrong with the PSMs and we should just remove them. And so what you could do, well, maybe I'll skip this exercise because we saw it quite a few times, is again, use filter features and filter for the mean SCR that is smaller than 10%. Here you can see also in some case when you divide things, well, you can get either missing data because there is missing data, or you can get infinite when you divide by 0. But anyway, those we want to remove too. So we apply this filter. Okay, so that's all for the PSM filtering. I hope it was interesting. So you can see, I'm pretty sure some of you might have other ideas. How could we filter PSMs? And I think there are like still great developments that we could do on how can we see which PSM do we keep, what's wrong, what's right. And I think this quality control at the beginning is very important and can still be improved, of course. Now we move to a first normalization. This normalization is maybe a bit brute in the sense that what we do is we divide all the samples by the reference sample. And this is done by the function called divide by reference. Again, it takes, we perform this on all the assays. To know which reference it is, it's just like compute SCR, is we look at the sample type. We take all the samples. Dot means like, it's like the white color, it matches everything. And our reference sample is the reference. And so like this, we divide all the quantification. It's like a first crude normalization, nothing very fancy. Okay, now it's interesting. So remember, this is all still at PSM. And now it's time now to move to aggregation. And so now we want to move from PSM to peptides. In this case, when they performed this, the authors, they actually simply removed duplicate PSMs to keep only one peptide. It's a bit weird. I mean, weird, maybe not weird, but it's not conventional. But I think this is an excellent way to show you how to apply custom functions. So remember, with QFeatures, we used call medians that was already available. But suppose you have like a brilliant new idea, you want to have some other aggregation function that you develop yourself. Well, you can provide it as a custom function. And so in this case, well, it's not fancy, but it's still custom. We call it remove duplicates. Then, so every assay that will be aggregated to a new assay. And so we need to provide new names for each of those new assays. And because I'm not very inspired, what I just did is I take the original name from the parent assay, and I just put peptides in front of it. So nothing very fancy. And so with this, we have all the information required to perform aggregation. The only thing, remember, with QFeatures, we were aggregating one assay to another. But in this case, we have more than one assay. We have many assays. So what SCP provides is a wrapper function to automate this through several assays. And this is why we call it aggregate features over assays, over different assays. So here's what I suggest you is to apply aggregate features over assays and to aggregate your PSMs to peptides. And don't forget to use the remove duplicates. Again, I'm just going to show the documentation. On the right. Oops. Yeah, maybe I already show the hint. So if you remember from the QFeatures, I mean, I went a little bit fast about it, but you need to say, okay, how does the function know that it has to aggregate? I mean, what should be aggregated to what? it does know it thanks to the fcall argument, and so actually what it expects is to say, okay, what's the grouping variable? And in this case, the grouping variable is the peptide name, and peptide name in this case is stored in the raw data as the peptide variable. I mean, very original. I mean, at least I hope this is clear. So it will automatically fetch peptide in the raw data, use it as a grouping variable for each assay separately. So actually, knowing this, we're almost there. Because, so we have aggregate features of assay. Our object is SCP. Which assays do we want to aggregate? Well, all of them. So we can use names. Wait, let me progressively show the answer up. Okay. So SCP, we want to aggregate over basically all assays. The fcall I just explained, well, it's peptide. So now how do we want to call those new assays? Well, we have stored something, we have stored all those names into peptide assays. This will contain all the new names. And the function is remove duplicates. So now if we run this, and here, yeah, probably one talking, your thing was still running. This function can take quite some time because we are aggregating over different assays. So again, if you want to ask some questions right now, we have still half an hour left, which I think is good. We can have, I think it should last for 5 minutes. If you can have a 5-minute question session, or if you want to get a drink, well, feel free. Wait, I see that. Yeah, Laurent, I see you've been answering quite some. You have been answering some questions. Are there things that I didn't cover?
And the last one was a general question about scope 2.
Okay.
But unless I'm mistaken, I think we can expect a substantial increase of aggregate.
Yeah, sorry. Indeed, sorry. Indeed, yeah, we were. So this is well, to go maybe a bit technical. There is something happening in S4 vectors when replacing an element in the list object. Well, which, like, if you scrap into those functions, you'll see it will happen somewhere. There are some checks that are probably not required and that take quite a long time. So there are some pull requests from Marcel that is pending, and probably that once that will be resolved, I mean, I hope it will get accepted. Well, once that's done, I think we'll get like something, about 20 time increase in time. So it will be 20 times faster than this. Just because here every time so you can see on the top, on the bottom left, it's aggregating function after assay after assay. And every time it's rechecking the whole data, the whole object, and which is maybe it is overkill, and avoiding this might increase. Can you speak about removing duplicate peptides when aggregating? Yeah, well, I went a bit quickly over it because I don't really wanted to talk about it, because I personally would not have done that, and it's probably this is pretty confusing. But well, I mean, this is maybe probably a good time to check it. So what it does, this function should expect a matrix, and so what we are going to do is we apply to that matrix for every column. We look at, okay, which one is NA, and well, it's not NA because we don't want. Well, if we want to remove duplicates, we don't want that we remove data but keep missing data. That would be not useful. So what we do is we take the first occurrence that is not missing. That's basically what it does. Wait, maybe let me explain this because I realize that's not clear. So what we do is we look. So for every column, so we look at the column and we look which one is not NA. From those that are not NA, wait, let me check. From the ones that are not NA, we take the first occurrence. And from that, we just take the first okay, for the first occurrence, we take that value in that column, and in the end, it will return simply, what will it return? It will return a vector of the same length as a number of samples. So for each sample, it will look, okay, it will just take the first non-missing data. That was a very long explanation, probably for not very much, but I hope it's a little bit clear. Okay, great. But yeah, I know I said it already, but I want to make very sure, don't use this for if you have your own data, or if you want to reproduce things, or you want to improve things, don't do it. Don't do it at home, please. It's not the right way to go. I would definitely go more for something like co-medians, or even better. This may be a good opportunity to tell it. If you go into aggregate features, so that's the function from QFeatures. Up you go here, and it will suggest you automatically to use the robust summary. It will take a little bit more time, but as it says, it's more robust. And yeah, it has been shown that using this function leads to better estimates of protein quantifications during aggregation. So yeah, so thanks for the remark. Okay, so it's finished. We have now aggregated all our different assays. And as I said, we had 24 assays. Now we made 24 new assays. So normally we should have 48. And indeed, we have 48. You see, now we have 24 starting with peptides. Okay, I'll maybe just skip this right now. The plot thing I'll show you afterwards because it will be nicer. Then there is another rather boring thing, is to clean missing data. So what we do is we replace all zeros. That can be either a true zero or a technical zero. And since we don't know, well, we prefer to just replace it by missing data. And later on, I think there are still great developments that has to be done for single cell. Is how do we estimate those missing data? How do we estimate that this is a true zero, or this is like a technical zero? And yeah, so that's one of the challenges that still remain. Yeah. So we do this basically using infinite is NA. So because we divided by the reference while some infinites came in. And we do the same, all the zeros are NA because what I just said. Now we have peptides for each batch, but that can, like, keeping all those assays separate might be difficult. And here, because we have peptides, several peptides are found across batches, so it may be better to condense this in a single table. We can do this using the join assay. Join assays, what it does, it takes again the QFeatures object. It asks for which assays do we want to aggregate. Those are the peptide. Do we want to join? Sorry, those are the peptides assay we just aggregated. And how do we call this new assay? We call it simply peptides. This takes some time. So now basically it takes the assay, the quantitative data, and then it will take the second one. It will match the peptide names, and you get bigger data, and then it will progressively expand that table into, so that until it gets all the assays that you want to join. And it does exactly the same for the raw data. So the raw data is kept. Okay, it's done. So now we should have one more, and we get, okay, you see 49 now. We have peptides. And now let's plot. You remember we could also plot a QFeatures object. Let's plot it. And here it's like, wow, okay. I mean, you can see the general what's happening. So we had our 24 PSMs that were now aggregated to 24 peptides, and those 24 peptides were all batches, and those are now joined into a single one. But you may say, we can't really read a lot here. So what we can do is use interactive equals true. And then you get actually the same, well, I mean, maybe not visually is not this, I mean, the idea is the same. Visually, it's maybe there are some things that change, but here you can now have it's a plot-like plot that is interactive. And so what you can do is then say, okay, I'm more interested by only those. Okay, now I can better read what is happening, and I can scroll back. I can fit the whole thing. I can look at something else. I mean, that's an issue when you have many assays. It becomes very hard to get a general overview of everything. But using this interactive plot, it will allow you to kind of, okay, look what you want to look and retrieve maybe one batch that you were interested, or how was it processed, and yeah, things like that. Okay, so now we are with our peptide data, and here I want to spend a little bit more time again. It's doing single cell quality control. I mean, that's again just like PSM. It's quality control is very important to make sure that your data that you are using is of high quality. So let's do it here on the cells. So in the scope 2 analysis, what they did was filtering the single cells based on the coefficient of variation, which is the standard deviation coefficient of variation is standard deviation divided by the mean. And something very, I think, useful with proteomics is that you have, because several peptides belong to the same protein, you can evaluate, you can estimate the variability of the quantification for a protein by looking at the variability of the quantification across the different peptides belonging to that protein. And so like this, you can have a coefficient of variation for each protein in each sample. Now, if you want to have an idea of how the average robustness of the quantification is across your cell, well, you can just take the average or the median. You can just take the median, and this will give you an idea of, okay, this in each cell, how robust are the quantification. And sometimes, I mean, there might be something wrong or that happened during sample processing, the preparation of the sample, that leads to highly variable cells, which you don't want to have. Okay, but then we need of course to have this information, this coefficient of variation, and this can be computed using the median CV per cell, so median coefficient of variation per cell. Wait, again, I don't want to. Okay, no, this maybe I'll just show you quickly how to do this. So you have this, you supply your QFeatures object again as usual. As usual, you point to in which assays do you want to compute this. Here we want to compute this in the separate peptides, because we will apply a custom normalization. So here, I mean, custom is not really custom. We coded it. We reproduced the normalization that they performed in the original article just by providing this argument. And then you need to say, okay, we want to compute coefficient of variation, but variation within what? Well, we want to group our peptides by the protein. So we want to quantify the variation of the peptides within a protein. Also, okay, you can have you can estimate a standard deviation or coefficient of variation, but like on two samples, on just 2 observations, that's probably not what you want to rely on. Here we say we want only coefficient of variation that are computed for at least 6 values, 6 observations. And then, okay, once that's done, it will compute the median CV across each cell, but where do you want to store it? And because we have data for each cell, naturally we want to store this in the cold data. And so that's why we need to provide a new cold data name. In this case, we call it median CV. So the computed CVs, median CVs, will be automatically stored in the cold data at the median CV variable. So let's run this. Oops. Yeah. And so here I included, so I take the cold data, and then I start to do some data filtering, and then I plotted. And I filtered only for 3 types of sample: macrophages, which are single cells, monocytes, which are single cells, and the blanks. And why the blanks? Because this provides actually a null distribution. So blanks, we know it's just what we find in blanks is what we don't want to see in a sample. And how does it behave? And then we can see here, so for instance, blanks, they behave like, well, they are here. We can see there are some issues here, and all those cells, they are better removed. So this is what you will do now. So I want you now, based on the cold data, to remove cells that are, wait, so yeah, here's the threshold. So here I've put arbitrarily the threshold here because I think it's a nice cutoff. So I want you to remove cells for which the median CV is smaller than 0.365. And how do you do this? You can do this because you're looking at the cold data, you can look at subset by cold data. And this function is actually provided by the MultiAssayExperiment, that's on which QFeatures rely. So again, let's look at the documentation. Oops. Subset by cold data. This is the one we want. So X, as usual, we always have this QFeatures object. Well, in that documentation, they call it MultiAssayExperiment, but since QFeatures inherits from it inherits from that function too. And then you need to supply the Y. And did I provide? No, I did not provide the hints. So maybe I can give a small hint. The hint is, so the Y should be a vector of logical thing. Okay, this true, this true, I want to keep it. False, I don't want to keep it. True, I want to keep it. False, I don't want to keep it. And so on. And this for every entry in the cold data.
I think I seem to have lost Christophe.
I think we lost you.
Okay, that's good. If you are still there, that's fine.
I mean, unless someone else wants to confirm it, I see you have like this low.
I also got the message that we lost Laurent briefly.
Okay. Maybe that's a good remark. If you have some issues, like if you feel it starts to lag or that your connection is quite weak, you can go in AirMeet on the bottom right. There is a little HD logo. You can click on it and you can go to LD. Well, you get lower definition, but at least it should be, the bandwidth should be reduced so you'll be able to get less technical issues. Wow, okay, this was just an info. Okay, let's move on to the solution. So, okay. There's part of the exercise I didn't mention. Okay, whatever. So probably what you did is you use subsets called data, SCP, and then you take SCP $operator to get directly access to cold data variables, and then median CV, that's the one variable that we just created with coefficient of variation. And we want it to be smaller than 0.366. Then what we also want is only to keep macrophages and monocytes, because at this point, blanks we don't care anymore, and all the others we don't care actually. So we just take those two. And so now we do this. And so I don't know if you remember, but each assay up to now, there were 16 columns corresponding to 16 samples per batch. And so let's wait a little bit for this. Yeah, great. See, now there is, well, now we removed quite a lot of samples. So now there's only 11. Yeah, you can see 11, 10. So we removed between 5 and 6 samples in the different batches. Okay, so that's it for the singles. Filtering on single cells again. Probably, and I mean, from RNA-seq, there are other very interesting metrics that you could use, like, for instance, the median intensity. In this case, in proteomics, it doesn't work. I mean, I'll be happy to explain this later on if we have time, but I'm afraid we don't have time. But you may think of any other metrics on samples on single cells that you could apply to get the best data out of it, and or at least remove the data that is not useful, or that could bias some analysis. Okay,
Christophe?
then. Oh yeah, sorry.
Just a quick note that we have 10 minutes left.
Yeah.
So
thanks a lot. Perfect. Yeah. So now there are a few steps that I'm gonna skip because they're quite again boring. So for instance, normalization. You can do normalization, divide your samples, divide your features depending on some statistics. And the idea is still the same. You provide QFeatures. You say which assay you want to apply, and then you apply whatever method you want. I'll suggest you to look in the documentation for all the available methods. You can use a sweep, which is a more flexible but maybe a little bit more complicated normalization function. Then we can also filter based on missingness. So for instance, we want only to keep in scope 2 they kept only features that have at most 99% missingness, which is a lot. Then we can do some log transformation. This is very trivial. And then we can do this aggregation of features. Wait, let me maybe just... yeah, maybe from now on, I'll just move to the compiled vignette. Just also for sake of time, I realize I'm running a little bit out of time. Oops. Where are we? We are at log transformation. So again, this is very trivial. Then protein aggregation. Here it becomes very similar to QFeatures because remember we have one peptide assay that we have transformed. So now we use the peptides log assay. That's one that was created just before. We aggregate based on protein sequence. We call this new assay proteins, and in this case, the author used the co-medians for aggregating. Again, some normalization, rather boring. Imputation, that's not boring. That's very important. But I think at this stage, imputation is still very basic, let's say, and I think some new improvement could be done to make some better imputation. And so, for instance, you can see here for each single cell, we can show what is the average missingness. So we can see we have between on average 65 and 70% missingness. Do we want to model this? Do we want to impute this? Yeah, this is something that still needs to be studied. In the case of the scope 2, they have used KNN imputation. Then, okay, here I want to spend I'll probably spend a little bit more time is doing batch correction. So next to imputation, batch correction is also a very important topic that requires still a lot of development and statistical, to have more thorough statistical description of what batch correction is and how to model it. In this case, in the analysis, the authors use combat, and yeah, we would not recommend combat. I mean, we still need to find the best method, and probably combat is not the best method. But just this is a very nice example of how can you apply your own custom function. How can you create your own custom function to make a new assay? Because combat is coming from another package called SVA. So what we need to do is extract the assay that we want to transform. So the latest assay is proteins norm 2, so normalized protein data contained in that assay. And we extract it using the get with call data. So it will not only extract just the assay, it will also transfer the call data from the QFeatures to the assay, so to the single cell experiment object. So in the end, we get a single cell experiment object. We can do different things. We can extract, here we needed some call data information from the QFeature that was transferred to the single cell experiment. We retrieve it from there. So basically the batch that we want to correct for, and then we have a model matrix. We provide it to combat, and we override the quantifications by this batch corrected quantifications. Okay, so that's very nice. Here you applied your function. It could be combat, but it could be anything else that you think is a good thing to do. Now we need to put it back into the QFeatures object. And this is performed using add assay. Well, the name is rather obvious. So we add assay, we we have our QFeatures object, we add the single cell experiment object, and we need to say, okay, this is the new name of our assay. So that's very nice, but if you do this, remember that QFeatures is able to memorize, or it stores the different links between previous assays. And here there is an obvious link with the previous one, that is that each protein matches to its own protein, and so it's a one-to-one relationship. So what we do is we do add assaying one-to-one from proteins norm 2. Remember that's the parent assay here, to proteins batch C. So that's the new name we gave it. And like so, we have added a new assay that has been transformed using a custom function. And with that, that's also, well, I mean, there's a last normalization step, but that's the main processing steps I wanted to show you. And now with the last 5 minutes, I just want to go very briefly into data visualization. Although I think we made a nice vignette about how to visualize data in QFeatures object, and here we will just have a quick look at it. So now, suppose you are very interested into filamin A. I took that one just as an example, but no particular reason. Filamin A is encoded as P213033. And we want to have a look at, okay, how was that? I mean, this protein seems to be very interesting. How was it processed during this whole vignette? So to do this, we want to apply ggplot. I mean, I do like ggplot, and what ggplot usually expects are long format object tables, but QFeatures is all but a long format object. So what we can use is the long format function. So now we have our QFeatures object, the final one, the one at the end. We can do some subsetting for that protein of interest. So remember, first index are for features. So we extract that protein and all the associated features that are to it. So all associated peptides and PSMs. And we can do this to get the quantitative data and the raw data. But we can also add some call data variables, and in this case, set, which is the batch. We can also add channel, which is in which channel was it found, and the sample type to have some nice colors coloring macrophages against monocytes. We do some filtering because we are interested. We saw there was something special in that batch, let's say, because, well, I don't know, there was some batch effect, or you think that batch might be more interesting than another. It's just an example to show you what you can do. And then we can supply it to ggplot, doing some facet wrapping, and get this kind of data where you have the data across. So here each facet is a different assay, and you can have, and each column here corresponds to a quantification channel, and you can see how the data went through the processing. So here we had our PSMs, we aggregated to peptides, and then we had our log transformation, and then we aggregated to proteins, and so on. And I think maybe the last message I want to give here is, like, look at this. If we look at our final data, so red are macrophages, blue are monocytes. Probably if we would have run a differential expression analysis on it, we would definitely see this as super differentially expressed. But when we look back at the peptides, and when we look back at the PSMs, we can see that we lost actually quite a lot of information by aggregating all of this into proteins. We had some variability here that we now miss. So it's very interesting. I think the QFeatures function, the QFeature class is very nice to show and explore this kind of effects, and is the ideal. And so this is my conclusion. And I think QFeatures and together with SCP for single cell application, it's the idea of framework to kind of study those kind of things, those kind of effects, and see what would be the best steps to apply to single cell data to in the end get the best downstream analysis or the best results out of it. And so, for instance, in my case, I would say I would rather try to apply some modeling on the peptides, or include the variability of the peptides before doing some aggregation where we lose quite some information. So I hope it was. Sorry. And last thing is just show you that because we're using single cell experiment objects, we can apply single cell. We can have an interface with single cell tools, and in this case, it's an example with Skater, when you can run typically a TSNE or whatever, or UMAP if you prefer, and see, okay, have a low dimensional view of your data and see that, okay, there seems to be some data that separates macrophages from monocytes. But with this, that concludes my our workshop. There are some further readings if you want, some vignettes. We also wrote a paper that you can access here. It's available on bioRxiv, and it will give you maybe a little bit more details on the technical aspects. And yeah, I mean, that's it. I hope you enjoyed it. If you have any questions, I mean, I don't think we have we have no time left, but I'll be very happy if you see me around on the tables. So I'll be very happy to answer any questions. And I would love to have some discussion if you have ideas for new developments, if you are into proteomics, or if you have ideas from how to transfer knowledge, basically. Thank you.
Thanks a lot, Christophe. That was great. And thanks also to everyone for joining and for following along. I saw there were many people who were following on the workshop on their own. So that's really great.
Nice.
So I think we'll wrap up here, and I'll see you around in the next session, which I think is the official welcome and opening of the conference. So thanks again, Christophe and Laurent, and thanks everyone for joining.
Thank you very much.
Thank you, Sophie.